This poster presents a cryo-transmission electron microscopy study on the visualization of aggregated superparamagnetic nanoparticles dispersed in a nematic liquid crystal matrix. The liquid crystals are a type of soft matter which exhibit orientational and/or positional order with maintaining fluid-like properties simultaneously. Nematic phase is the most simplest S phase, where rod-like molecules are locally aligned roughly along the same direction, and there can be topological defects where domains with different alignment orientations form discontinuous boundaries. Many works have been performed to see what happens if metallic nanoparticles are doped with the nematic LC materials with respect to the development of optical devices. Especially, functionalizing the LC by mixing small amount of superparamagnetic nanoparticles has been attracting interest for future applications. However, fundamental investigations are still lacking on how doped nanoparticles behave in the anisotropic host media. As shown in the first figure in the background, it has been well known that the nanoparticles tend to localize at the loci of topological defect in the host LC matrix. Nevertheless, there is no direct observation on the shape and structure of the localized and aggregated nanoparticles at the defect sites. In order to achieve this, Cryo-EM is the best method for the direct visualization because the intrinsic sample properties can be fully maintained using cryo techniques. As is seen in the second figure in the background, metallic nanoparticle clustering structures in the aqueous solutions have been already imaged by applying Cryo-EM methods. What we have done for this was to form vitrified nematic LC films in a microperforated carbon layer that caused the TEM sample grid. After treating the holy carbon grid with plasma to avoid particle absorption on the carbon layer, the LC spion mixture was loaded on the grid and quickly blotted. The specimen was then directly plunged into a cryogen to finally form a thin vitrified LC film where the spions were trapped. The specimens were subsequently transferred to a cryo holder and were observed in a TM at 200 kilovolts under low dose of electron beams. Thanks to the strong electronic contrast of the metallic atoms in spions, we could directly visualize the aggregation structure of nanoparticles doped in the nematic media. As is seen in the micrographs in the results and discussion, the aggregates basically had sphere-like morphology, and the berry-shaped unit aggregates combined into elongated and fractal-like structures that spanned a few hundreds nanometers in length. In a sphere-like unit aggregate, 30 to 40 spions agglomerated with a surface-to-surface distance of 1.9 nanometer, which exactly corresponded to the chain lengths of Oleg axes that were coated on the 5.8 nanometer sized spion. Therefore, we conclude that the spions were close packed within the unit aggregate. Our research promotes the fundamental understanding on how colloidal nanospheres behave in an anisotropic fluid. Furthermore, The data have a potential to be used for designing novel hybrid materials and devices that combine properties of liquid crystals and magnetic particles.